Idiopathic pulmonary fibrosis is a chronic and progressive lung disease. Diagnosis and prognosis is very poor. The mortality rate is very high. Patients, they survive about 3 to 5 years after diagnosis. Currently, treatments are few. There are only a few drugs approved by FDA. So there is a need for to identify novel targets, new therapies for cure or ameliorating the disease or whatever. So my laboratory back in the US has been interested in looking at a lot of lung pathologies, including idiopathic pulmonary fibrosis or IPF. To study the human disease, we need animal models, and there are 2 animal models that are currently used to study IPF. One is the bleomycin inflammatory model, and the second one is the radiation-induced lung fibrosis model. So we have used both the models in our study to understand some mechanisms by which the process of fibrosis is initiated, probably in the humans. By studying these animal models, we should have some better understanding of the mechanisms, and so that we can put forth some hypothesis to see how that works in the humans to come up with better drugs. So we have been doing work in the area of bioactive lipids. These are lipids that are generated in the tissues and in the fluids of the humans under various pathologies. And specifically, we have been interested in looking at 2 bioactive lipids. One is lysophosphatidic acid, and the second one is sphingosine-1-phosphate. These are naturally occurring bioactive lipids that are found in our tissues, in the plasma, and in circulation. And they do have a role in the pathology, and especially in idiopathic pulmonary fibrosis. A paper published in Thorax by another group from Italy in 2012 reported that sphingosine-1-phosphate, the bioactive lipid, is elevated in the BAL fluids from patients, IPF patients. And they also mentioned in the paper that there was an increase in the expression levels of proteins that are involved in the synthesis of this particular bioactive lipid. So we were interested, since my lab is interested in sphingosine-1-phosphate, we were working on the bleomycin model as well as the radiation model, and we took this study to a step further to better understand what is the mechanism by which this sphingosine-1-phosphate is generated, why its levels are increased in idiopathic pulmonary fibrosis patients in their BAL fluid, and what are potential mechanisms that we can look into that modulate the S1P levels in the animal models. There are 2 enzymes that specifically regulate sphingosine-1-phosphate. One belongs to the enzyme that synthesizes this lipid from sphingosine. These are known as sphingosine kinases. 1 and 2, there are 2 isoenzymes. And the other set of enzymes are catabolic enzymes, or enzymes that degrade sphingosine, that is sphingosine-1-phosphate that is generated. And specifically, we were interested in looking at sphingosine-1-phosphate lyase. In idiopathic pulmonary fibrosis, also in the animal models, both these enzymes are upregulated. Sphingosine-1 kinase that generates S1P, that's why there is more S1P in the IPF patients. And interestingly, the sphingosine-1-phosphate lyase is also upregulated. So we were interested why the enzyme that is degrading this lipid is also upregulated. And our studies suggest that possibly the lung tissue where the fibrosis process takes place, there is a balance between the synthesis mediated by these kinases and the S1P lyase that degrades this enzyme. So lung has a natural mechanism wherein there is a balance between the synthesis as well as the degradation. And by degrading this lipid by this enzyme, the lung is able to protect the tissue from the fibrotic process. So we have developed cell culture models using lung fibroblasts from IPF patients as well as lung fibroblasts from animal models to understand the mechanisms by which one of the cytokines known as transforming growth factor beta or TGF-β that is involved in the pulmonary fibrosis. So TGF-β in lung fibroblasts actually upregulates S1P lyase, the enzyme that degrades S1P. And this upregulation of S1P lyase actually decreases the S1P levels, in turn increases S1P in addition to sphingosine kinase, and affects a process that is known as autophagy in the cell. Autophagy is a natural process wherein cells can degrade macromolecules that are generated inside the cell. These are proteins, lipids, DNA, and so on and so forth. And the sphingosine 1-phosphate that is upregulated in IPF actually downregulates autophagy. And it has been published earlier that autophagy is actually good for IPF. So if the autophagy is higher, then fibrosis would be probably lower. And in this particular case, since sphingosine 1-phosphate downregulates autophagy, it is actually bad in IPF. So the sphingosine 1-phosphate lyase that degrades S1P would be beneficial and increase autophagy. So that's our working hypothesis right now, and so we are testing this hypothesis in animal models as well as cell culture systems. And our ultimate goal here would be to develop small molecule inhibitors or activators. And in this particular case, inhibitors for sphingosine kinase 1. By blocking sphingosine kinase 1, we would lower the S1P levels, or by activating S1P lyase, also we will be lowering S1P levels. So the ultimate goal is to lower the S1P levels in idiopathic pulmonary fibrosis, increase autophagy, and thereby decrease fibrosis.